MCQ 31 have us look at the relationship between polypeptide and DNA structure. So again, as the summary goes, the central dogma of human biology itself will state that we started off by having DNA, and we will undergo the process of transcription, maintaining the original copy of our DNA to get a single-stranded script known as mRNA. And mRNA will undergo the process of translation, and this is with the assistance of a few other items inclusive of amino acids, and amino acids are transported towards the mRNA via tRNA, so long that the codon on mRNA is complementary to that of tRNA. It will bring a specific amino acids, and the reading of the mRNA is facilitated by the ribosome. So the end result of translation, this will yield a polypeptide, and the polypeptide could be sent towards the Golgi apparatus for post-translational modification for events such as movement out of the cell. But this is not something that we're going to look at today. So DNA, we'll have it as double-stranded. So based on the strands itself, if we're going to select one of the strands for the purpose of transcription, let's say the strand that's at the bottom over here, the bases on it could either be A, T, G, or C. Now that selected strand will undergo transcription to give us a single-stranded mRNA. So each 3 bases on the mRNA is described to be as a codon, and they can only have bases such as adenine, uracil, guanine, or cytosine. So the codon itself will respond with tRNA. Now tRNA will have 3 bases at the bottom here that is bringing along an amino acids. That 3 bases at the bottom that we're seeing here is known as the anticodon. So again, depending on which anticodon, the amino acids that's attached towards the tRNA is different. So the sequence of the mRNA will summon different forms of tRNA and therefore brings different sequence of amino acids to form the polypeptide. So looking at the option here, the sequence of amino acids in a polypeptide determines the sequence of bases in DNA. Now this is wrong because this actually has to be the other way around. DNA determines the sequence basis of my mRNA, which then determines the sequence of amino acids on my polypeptide. In option B, the sequence of amino acids in DNA. Now already this is a red flag. DNA does not contain amino acids, but rather deoxyribonucleotide. In part C, the bases in polypeptide, this is also wrong. The base is found in mRNA or DNA, but not in the polypeptide. So lastly, the sequence of bases in DNA determines the sequence of amino acids in the polypeptide is correct. We can add in a furthermore distance. The bases will determine the bases in mRNA, which then determines the tRNA that we're going to be using, which ultimately determines the sequence of amino acids in the polypeptide. So the answer for 31 is D.